research involves following the methods previously used in the Husain Lab to find out how the host gene, EP300, is either helping or hindering the replication, growth and release of Influenza A virus in infected human lung cells. I would like to say a huge thank you to the Otago Medical Research Foundation and the Stonelake Foundation for funding this opportunity for me, Dr. Matloob Husain, my supervisor, and Farjana and Bennett, the PhD lab students who have dealt with me over the past few weeks. Influenza A virus, which infects the upper respiratory tract, is responsible for many devastating pandemics in history, not to mention its yearly seasonal epidemics. Because of this historical pattern, it's well known that influenza A has the possibility to cause a future pandemic of catastrophic proportions, perhaps even worse than the one we are currently facing. This is particularly worrying in the current situation we're facing with COVID-19. Because of this threat, our lab's research focuses on understanding how host genes, or instructions to make protein inside of cells, can increase or decrease influenza A virus's ability to infect human lung cells. We do this by performing a knockdown of the genes and seeing if the amount of replication, growth and release of influenza A virus increases or decreases with respect to a decreased amount of that gene in the cell. I like to think of the project like this. Think of a human lung cell as a small town with a graffiti problem, the graffiti representing an infection with influenza A. The amount of graffiti normally looks like this when a guy called HDAC6, a gene, is present at his normal level. The Husain Lab previously sent out Constable HDAC6 siRNA, who magically decreases the height of HDAC6 specifically, or the protein expression, significantly altering his ability to function normally. When they did this, the amount of graffiti increased significantly, indicating that normally HDAC6 is acting as an anti-graffiti, or antiviral. My summer project is working with EP300 hat gene, and I will send in Constable EP300 siRNA to decrease the height of specifically EP300, decreasing his ability to function normally. Based on pre-existing data, when I do this, I expect to see the level of graffiti decrease, indicating that normally inside of the cell, EP300 is pro-graffiti or pro-viral.